Hello friends, welcome to BMH Learning. This video deals with replication fork stalling. First, let's know what is a replication fork. The figure shows a replication bubble, which is an unbound and open region of a DNA helix where DNA replication occurs. The two sides of each bubble, where it goes from zipped to unzipped, are called replication forks, and from these two sides the replication is carried out. Sometimes, due to certain mutations or any other cellular blockage, the replication fork is halted and cannot go forward. This situation is referred as stalled replication fork. There are various potential pathways to repair and restart a stalled replication fork. As you can see in the diagram, parental DNA strands and newly synthesized DNA strands form a stalled fork due to the presence of DNA damage or replication blockage. Now, depending on the cellular response and conditions, one of the following way can be employed to reverse the stalled fork situation. In the classical model, single-stranded DNA at a stalled replication fork is bound by a replication protein A, that is RPA. RPA then recruits ATR to activate the DNA damage response. Now, ATR-dependent DNA damage response stabilizes the stalled replication fork. Following ATR-dependent stalled replication fork stabilization, the damage or blockage is repaired either by DNA lesion bypass, direct damage removal, or template switching via fork regression. This was all. Thanks for watching.